From the moment we're born, our bodies are a marvel of biological activity, constantly adapting, repairing, and growing. But what happens when that incredible machinery finally stops? What's the science behind the ultimate transition we all face? Today, we're going to embark on a fascinating, educational journey, not into the realm of horror, but into the intricate biological processes that unfold in the human body during the first 72 hours after death. It's a scientific timeline, step by step, explaining the why behind each profound change. The journey begins at the moment of clinical death, which many might consider the official end. This is when the heart stops beating, and breathing ceases. Without these vital functions, oxygen no longer circulates to the brain and other organs. While the heart and lungs have stopped, this isn't the absolute end for every cell. In these initial minutes, there's a window where resuscitation might still be possible because not all cellular activity has completely ceased. It's a crucial distinction between the cessation of outward life signs and the deeper, cellular processes. Even after clinical death, the brain doesn't immediately go silent. For a few precious minutes, some residual electrical activity can still be detected. Think of it like a light bulb slowly dimming rather than instantly extinguishing. However, without a constant supply of oxygen and glucose from circulating blood, these electrical signals quickly fade. Neurons, the brain's specialized cells, are incredibly sensitive to oxygen deprivation. This brief period of residual activity soon gives way to complete neurological silence, marking the brain's definitive shutdown. Once the brain goes silent, the lack of oxygen and nutrients impacts every cell in the body. While clinical death is immediate, biological death, or cellular death, is a gradual process. Without oxygen, cells can no longer produce ATP, the energy currency they need to maintain their delicate balance. This energy failure means cell membranes can't hold their integrity, and internal pumps stop working. Enzymes, which are usually contained within cellular compartments, start to leak out and begin to break down the cell from within. This process is called autolysis, or self-digestion, and it's happening everywhere. One of the most immediate and noticeable changes after death is the drop in body temperature, a phenomenon known as algor mortis. When living, our bodies constantly generate heat through metabolic processes. Once these processes cease, the body begins to lose heat to the cooler surrounding environment, much like a cup of hot coffee gradually cools to room temperature. This cooling is not uniform and depends on several factors, but it's a predictable physical change that forensic scientists use as a crucial indicator. The rate at which the body cools isn't linear. Initially, the body loses heat more quickly, then the rate slows down as the temperature difference between the body and its surroundings decreases. On average, a body cools at about 1.5 degrees Fahrenheit, or 0.83 degrees Celsius, per hour during the first 12 hours. Factors like the ambient temperature, clothing, body mass, and air currents all influence this rate. A body in a cold environment, like snow, will cool much faster than one indoors or heavily clothed. This makes algor mortis a vital clue for estimating the time of death. As the heart stops pumping blood, gravity takes over. The blood, no longer actively circulating, begins to settle in the capillaries and veins of the body's lowest-lying areas. This causes a purplish-red discoloration on the skin, a phenomenon called livor mortis or lividity. Think of it like sediment settling at the bottom of a still pond. The blood pools, and hemoglobin, the oxygen-carrying protein in red blood cells, breaks down, releasing its pigment and creating this characteristic staining. It typically starts to become visible within 30 minutes to an hour after death. Initially, livor mortis is blanchable, meaning if you press on the discolored skin, the color temporarily fades. However, after about 8 to 12 hours, the capillaries rupture, and the blood cells break down completely, staining the tissues. At this point, the lividity becomes fixed and will no longer disappear when pressed. This fixed pattern tells forensic investigators if a body has been moved after death. If the lividity is on the back, but the body is found face down, it indicates the body was moved, providing critical information about the circumstances surrounding the death. Perhaps one of the most widely known post-mortem changes is rigor mortis, the stiffening of the muscles. In life, muscle contraction and relaxation require energy in the form of ADP. After death, cells stop producing ADP. Muscles use up their remaining ADP, but there's no new supply to release the contraction. Without ADP, the muscle fibers, specifically actin and myosin, remain locked together, causing the muscles to become stiff and rigid. This isn't an active contraction, but rather a permanent state of tension due to biochemical changes. Rigor mortis doesn't affect all muscles simultaneously. It typically begins in the smaller muscles of the face and jaw within 2 to 4 hours after death, then progresses to the larger muscles of the limbs and torso over the next 4 to 12 hours. The body usually reaches its maximum stiffness, or full rigor, around 12 to 24 hours post-mortem. Like algor mortis, the rate and extent of rigor mortis are influenced by factors such as ambient temperature, activity before death, and body mass. A body in a warmer environment will develop rigor faster. Interestingly, rigor mortis is not a permanent state. After reaching full stiffness, it eventually begins to dissipate, usually starting around 24 to 36 hours after death and completely fading by 48 to 72 hours. This relaxation occurs because the cellular enzymes, which we mentioned earlier with autolysis, continue to break down the muscle fibers themselves. As these proteins degrade, the locked connections between actin and myosin are destroyed, allowing the muscles to relax once again. The body returns to a flaccid state, signaling further decomposition. While rigor mortis is progressing and dissipating, the internal process of autolysis, or self-digestion, continues relentlessly. Lysosomes, which are sacs of powerful enzymes within cells, rupture and release their contents. These enzymes, designed to break down waste products and old cell parts in life, now turn on the cell itself. This internal breakdown is particularly evident in organs rich in enzymes, like the liver and pancreas, which start to soften and lose their structural integrity relatively quickly. It's the beginning of the body breaking itself down from the inside out. As autolysis weakens tissues, a much larger force comes into play: putrefaction. This is the stage where bacteria, particularly those residing in our gut, begin to multiply unchecked. In life, our immune system and physiological barriers keep these bacteria in check, but after death, they have free rein. They start to digest the body's tissues, breaking down complex organic compounds into simpler ones. Putrefaction is characterized by a distinctive color change, odor, and the production of gases, making it the most obvious sign of decomposition. One of the earliest external signs of putrefaction, usually appearing within 24 to 36 hours after death, is a greenish discoloration of the skin, particularly over the lower abdomen. This color change is due to the breakdown of hemoglobin by bacterial enzymes and the formation of sulfhemoglobin. This compound gives off a distinct greenish hue as it reacts with hydrogen sulfide produced by the multiplying bacteria. As decomposition progresses, this discoloration will spread across the entire body, turning it into a mottled, greenish-black appearance. As bacteria continue their work, they produce various gases as byproducts of their metabolic processes. These gases include methane, hydrogen sulfide, carbon dioxide, and ammonia. Trapped within the body's tissues and internal organs, these gases begin to accumulate, causing the body to swell and bloat. This bloating is most noticeable in the abdomen but can affect the entire body, making it appear distended. The pressure from these gases can become so intense that it can force fluids and organs out of orifices, or even rupture the skin, creating gruesome effects. Along with the visible changes, the microbial activity during putrefaction also creates a very distinct and powerful odor. This smell is primarily caused by volatile organic compounds, notably putrescine and cadaverine. These chemicals are the result of bacterial breakdown of amino acids like lysine and ornithine. The scent is often described as sweet but overwhelmingly foul, attracting insects and scavengers from a considerable distance. It's a primal signal that decomposition is in full swing, a natural chemical signature of death. All these post-mortem changes, algor mortis, livor mortis, rigor mortis, and the early signs of decomposition, are incredibly valuable to forensic scientists. By meticulously observing and measuring these phenomena, investigators can estimate the post-mortem interval, or time since death. No single factor provides an exact time, but by considering all these indicators, along with environmental conditions, a fairly accurate window can be established. It's a complex puzzle where each biological change serves as a critical piece, helping to reconstruct events and bring clarity to investigations. While our focus today has been on the first 72 hours, the process of decomposition doesn't stop there. Beyond this initial period, the body continues its journey of decay. The bloating becomes more pronounced, skin slippage occurs, and insects like flies and beetles lay eggs, accelerating the process. Eventually, soft tissues fully liquefy, leaving behind bones. This later stage, known as active decay and advanced decay, involves a complex ecosystem of organisms breaking down the remains until only the skeletal structure is left. It's a continuous, natural recycling process. What happens to our bodies after death is not a morbid end, but a fascinating series of scientific transformations. From the last electrical spark in the brain to the onset of microbial activity and gas production, each stage is a testament to the intricate biological processes that govern life and death. It highlights how connected we are to the natural world, even in our final moments. Our bodies, once complex living systems, become part of the grand cycle, returning their constituent elements to the earth to fuel new life. It's a humbling and awe-inspiring spectacle of nature. Understanding these post-mortem changes not only satisfies our curiosity but also provides vital tools for forensic science, helping to bring clarity and justice. It reminds us that even in death, our bodies tell a profound story, rich with scientific detail. We hope this educational journey through the first 72 hours after death has given you a new perspective on these natural, universal processes. If you found this explanation fascinating, please give this video a thumbs up, subscribe for more scientific deep dives, and share your thoughts in the comments below. What other biological mysteries intrigue you?